So, good morning, everybody. We are going to start with the first keynote of this morning. Just before I start, let me tell you that for Julio to hear your questions, the only way to do it is through a microphone, so I will have to handle with this micro, and then you will have the questions, okay? So it's a pleasure for me to introduce the keynote speaker of today, Dr. Julio Saez-Rodriguez. He did his PhD in chemical engineering, then the postdoc in MIT and Harvard, and became PI in the MBA in Cambridge. Then he moved to Aachen, where he was a professor of computational medicine, and he works now in Heidelberg, also as professor of medicine bioinformatics. He's an expert on the study of functional networks and signal networks and the role in these modifications affecting on diseases. So thank you very much, Julio. The floor is yours. I close now the micro.
Thanks for the very kind introduction, and it's a pity I cannot be in person with you guys in Barcelona. So, but yeah, I'll try nevertheless to share with you some of our work in the next hour or so. And yeah, so we are in Heidelberg and in the medical faculty, and I should disclose the funding from GSK and Sanofi, and since it's from Robert and Aztecs, two small startups. And with that out of the way, let me first start at a bit of a higher level of what is our broad interest, and then trying to go a bit deeper also towards how, at least in our group, we try to combine both a more data analysis, bioinformatics, machine learning, and strategy with a complementary dynamic mechanistic modeling. And I hope these strategies and methodologies can be interesting for you, and of course later we can discuss in more detail. So generally speaking, we try in our group to see how we can use different large-scale omics data to better understand and better predict which therapies are going to be working for a particular patient. So as you know, we can generate with increasing accuracy and scope and lower price different omics like transcriptomics, proteomics, and so forth. So these are measurements of these different molecules across many, if not all, genes. And then for different samples, the samples can be actual patients, or can be different model systems for the labs, and so on. And also something, and I will come to this later, is it's becoming now also feasible to measure this not only for a particular sample, but in that sample to measure all these technologies or these omics for each individual cell and in tissues to do this with spatial resolution. And as I said, what we try to do is to try to use this data and also a bit even the measurements of the microbiome, so not only on the patient, but also the different microbiomes that are present sometimes in different tissues, to understand and predict which therapies will work better for the different patient. So if you think about how to go about this question, a very natural way is to say, okay, let's take all this large data and feed them to our favorite statistical or machine learning method, and then try to extract relationships between the molecular features and, for example, the prognosis of a disease, so it's a particular molecular biomarker of disease, or build a predictive machine learning model that will tell us if, let's say, a drug is going to be effective in a particular patient or not. And as a model system to study this, we and other groups used large collection of cell lines. So this is around 1,000 cell lines. There is a large collection at the Sanger Institute led by Matthew Carney. There is an equivalent one at the Broad Institute. And so all these cell lines, they come from different disease of origins, so they represent different tumor types or individuals that have different tumor types. And then we have different of these omics data, like transcriptomics, genomics, and so on. And this keep being added, so now it was released, for example, the proteome of the cell line. So you have a lot of these molecular features, and then all the cell lines have been treated with drugs. It's around 400 different drugs, but also now more and more in drug combinations. And then what then you have is for each cell line, each of these drugs, and you have like a drug response curves. So increasing amount of drugs, seeing how many of these cancer cells survives. And this is typically summarized by the IC50, how much drug you have of the cells. And then, as I said before, so you could just try to use machine learning, input your data into the algorithm, and try to predict efficacy of the drugs. And we and many others have looked at this and try many different algorithms, from very simple linear models to more advanced Bayesian technologies. And when you do this, and I will just not get into the details, I'm trying to describe this to make the point that in general, and in this particular case, using machine learning, even if it's very advanced methods, and even in this relatively well-defined context of cell lines from the laboratory, the algorithms work to some degree, but in many cases, the predictability is very low, so they don't really are able to tell us whether a drug is going to work or not. And even more, the interpretability is limited. So out of these algorithms, it's known that they're often like black boxes. So even if we can predict well whether a drug works or not, it's very hard to understand why, what's the molecular mechanisms behind this. And for this reason, in our group, we have a focus on how we can help machine learning using biological knowledge. And I will show you in a moment what I mean by biological knowledge, but this is different molecular data, pathways, and so on. And the idea is that rather than feeding into your algorithm or your omics data, you use the prior knowledge to extract signatures, features such as the activity of a pathway. And then by using this instead of the raw data, you have a twofold benefit. First, you don't need to look at all genes. Instead, you look at maybe dozens of features, and having less input features, less dimensionality, increases the power of the statistical methods. And then, because these features are rooted on cellular processes such as pathways and so forth, they are mechanistically interpretable. And all the tools and resources I will show you today are freely available in our website. So if anything of this is interesting, you're very welcome to try and to reach out to us if you have any questions. The first aspect one needs if you want to use prior knowledge is to have access to it. And to facilitate this process, we developed a resource called OmniPath, which is simply one-stop shop which gives you access to many different resources. So over the years, we built different types of knowledge, starting from pathways and networks, adding information about complexes, annotations such as localization of proteins. And more recently, in the context of single cell data, and I will come to this later, we have also included information about cell cell. The total is over 2 million annotations for over 20,000 proteins. And all of this is available either through bioconductor R package in Python module or Saez-Rodriguez plugin. And so the basic idea then behind OmniPath is that you have access to many different resources, and you can take the one that is more suitable for your analysis in the same way that if you wear glasses, you try different lenses until you find the right to see the best. And with OmniPath, you look at different resources and you pick the right one or combinations of several to better analyze the data. So once we have this prior knowledge, as I said before, we try to extract mechanistic features. And in our hands, a very valuable strategy to extract features is to think of omics data as the downstream or the footprint of a process of interest. And I will elaborate this with some cases, but the general idea is that I'm interested to know what's the activity of something like a pathway, and these are often hard to measure directly, at least at high throughput. So instead, we take the omics data that is easier to measure, and if we know how the process of interest affects the omics data, we can estimate the former from the latter by using causal links that are coming from the biological. To be a bit more specific, let's say you have transcriptomics data, so you can use this to estimate the activity of a pathway. And the idea here is that if I want to know if this pathway here, that is shown in magenta, is active, instead of looking at the expression of the genes in the pathway, I would look at the expression of the target genes, shown here at the bottom. because the fact that there is more expression of the genes on the pathway doesn't mean that there is more proteins. So these blue nodes here, the contiguities of the pathways are proteins, are not transcripts. So more transcript doesn't mean more protein, and more protein also doesn't mean more activity. So this depends also on, for example, if the protein is phosphorylated or if it's in the right place, which is in this case in the nucleus. Then this is what we use to estimate pathway activity. We can use the same to estimate the activity of transcription factors. And again, the idea is that it's not looking at the expression of transcription factor itself. We look at the expression of the target genes upstream of it. And then what we can do is, if we have estimated, in this case, transcription factors and upstream pathways, we can try to identify key pathways that paths that connects them in a large signal network, as is shown here in orange. So in a bit more of detail, this is a tool called Carnival that we take from OmniPath, a large network. Here you study a small one, but it's a network of hundreds or so of different nodes. If we have transcriptomics, we use it to estimate transcription factors and the upstream pathways. And then an algorithm tries to find the causal paths that explain how changes in upstream pathways define the activity of downstream transcription. And going back now to the cell lines that I showed you in the beginning, now we can try to go back to the data and try to get a bit more of mechanistic insight of what's happening. And then what we do is, because from all of this around, as I said, that goes online, we have the transcripts, but we also have the DNA. We can use the transcripts to estimate transcription factor and pathways, and actually the DNA, we can map them on the network to include the effect of different mutations. And then we can build for each of our 1,000 cell lines a different network of causal paths. And then we can try to relate these causal paths to the drug response data. So we can try to see if there are specific components whose activity we have inferred from this network, and try to see if those are correlated with drug efficacy. And in this way, try to find biomarkers or predictors of drug response that are coming from the network analysis, not simply sort of from the actual level of the particular things. And I will not go into the results into detail, but what we see is that in fact these network approaches can find new biomarkers, new predictors that we would not see when looking only at the data. So until now I talk about transcriptomics, but the same ideas can be expanded to other type of omics. So if you have phosphoproteomic data, this is phosphorylation of proteins, and this is mediated by kinases. So the same idea as before applies. I can estimate the activity of kinase by looking at the changes in the downstream phosphorylations here in green. Or if I have metabolomics here on light blue on the right, I can use it to estimate the activity of metabolic enzymes. And in all cases, it's the same concept that I look at changes in the footprint in the omics data to find the activity of the upstream nutrients. And then what I can do is, in this multi-ohm context, I can apply again this idea of finding the causal path in the network that I showed you before with this tool Carnival. So now applied to multi-omics, this a method called Cosmos that we recently developed. And I will tell you a bit more about this, and I will do so in the context of specific application. So it's a study where we look at kidney cancer. So colleagues Dr. Kramer, Christian Fresa, and Jessica Olsen. We had samples of kidney cancer and the matching of the tissue. So using mass spectrometry and RNA-seq, we obtained transcriptomic, phosphoproteomic, and metabolomic data. First, we applied what I just described. So from the transcriptomic, we estimate transcription factor activities, from the phosphoproteomic kinases, and from the metabolomic enzymes, here on the right. And then using this Carnival algorithms, we try to identify the key pathways that connect. So when you apply this to the real data, starting point, this is, as I said, this is very large networks that combine signaling, but also metabolism and molecule protein interactions. So it's a huge kernel, as you can see here, from 170,000 interactions, and there is not very much you can do with this very complex network. But now, if we go and remap the activity of this transcription factor, kinases, and metabolic enzymes, as described in the previous slide, And then you find in this large network which pathways are connecting them. So how can we explain that, for example, in cancer, a particular kinase goes up and then a particular transcription factor is going down? So the resulting model, shown here on the right, is still quite complex, but much more manageable than the large network. So it has around 400 interactions, as you can see. And then you can really zoom in into these components and identify specific pathways or processes that are deregulated in the context of kidney cancer. And these are bridging from signaling to gene regulation to metabolism. Here we highlighted in 5 mechanisms that either for some of them there was supporting literature that they were indeed important. Some others are hypotheses for new analysis. And then any of these components, in principle, if it's a key regulated process in kidney cancer, in this case, are potential candidates for therapy. But at this point, I like to emphasize that all these methods really they only give you hypotheses, so it's not like ground truth. It's not for certain that what the method predicts is real. So all these are hypotheses that needs to be further validated in orthogonal strategies, perhaps with dedicated experiments. So I'll summarize then what I have shown you so far. So this idea that you have omics data, you can first extract the features, the signatures, and in our case, using this idea of the footprint to look at the changes in omics data as a way to estimate the activity of upstream process. And one thing that is important to note is that these strategies is robust to missing data, meaning that because the footprint of, for example, a pathway maybe it has 100 different genes, even if you don't measure all those genes, the method still works because you have enough to get signal. I will come back to single cell later, but the same, for example, is for phosphoproteomics, but it's also not complete. And then this footprint or footprint-derived features, we can map them on large networks and use that to extract key mechanisms that, as I said before, are hypotheses for further validation. So I said this before, I want to emphasize this again, that the methods that I just showed you work for bulk, but also work for single cell. And in this study here, I will not go into details with a number of benchmarks to show that you can apply this to single cell. And so then what this means then is that if you have single cell data that is very popular these days, you can use this resource, OmniPath, and the tools I showed you, but also other tools to try to estimate from single cell data, activity of pathways, transcription factors, and so forth. And because OmniPath has access to many different resources, we can also study cell-cell communication and use connect to external tools such as NIST, which is very common using single cell data. But let me tell you a few more things about this cell-cell communication, because this is something quite broadly used for single cell data. So in case you haven't looked at single cell data, what many people do is to say, okay, if I have single cell RNA, I use it to cluster groups of cells. And then I want to know if these groups of cells, that can be, for example, different cell types, like macrophages and fibroblasts, for example, you want to know if they are communicating, if they're exchanging information. And then what you would say is, okay, if a group of cells have a high level of expression of a particular ligand, that would be here this yellow triangle, and the other cell type B has a high level of the corresponding receptor, it is likely that these two cells are communicating. No? But of course, similarly to what I said when I described the pathway activity, the fact that there is more RNA doesn't mean that there is more protein, so it doesn't mean really that there is communication. So these methods are all indirect. And there is many methods, which all follow the same principle, but they have different set of distributions. So it's a way to kind of allow us to combine, compare, and understand all these different methods, building on OmniPath, where we have access to all those different databases. We built a tool called Liana, which allows this analysis framework for this ligand receptor interaction, and combines many resources, many of the different methods. And then, as I said before, by inputting a single cell RNA in different clusters, you can estimate cell-cell communication. And in a paper that is coming out soon, and I guess if you want to know more, let me know. We benchmarked many of these methods and resources, and the summary is that we found that most of them are able to capture some signal about cell-cell communication, but it's far from perfect. And this makes sense if you think about this biologically. So as I said before, what these methods do is to combine the expression of the ligand in a particular cell type with the expression of the receptor in another cell type. But in truth, communication is mediated by proteins or other molecules that are not RNAs. So in these methods, you're assuming that from the expression level, you can estimate the whole process, but of course, after the RNA expression, translation has to happen into protein, then this protein has to be secreted from the cell into the medium, it has to be fused, it has to reach the target cell, and then trigger a response. And there is a lot of steps that are not really captured by single cell RNA data, but nevertheless are certainly happening. So that's why, as I mentioned before, we need a number of benchmarks to try to understand how well these technologies that they don't really capture all these intermediate steps, how much they can approximate. And it's very hard to benchmark this. There is not really good ground truth, but we use indirect measurements, such as spatial transcriptomics. And in general, as I said before, we find that many of these methods get signals, so they capture likely signaling events, but not of them are right. So similarly to what I said before when I was talking about network analysis, all these bioinformatics methods, they only give you hypotheses, but then you should further investigate and validate with independent methodologies. And so the last frontier or dimension that is very important in this context of omics data is spatially resolved data. So single cell data is very exciting. You can look at individual cells, their molecular profiling, but for doing so, you have to dissociate them. And if these cells come from a particular tissue, so you don't know where these individual cells were in the tissue. But some technologies, and this field is developing as we speak very rapidly, allow us to not only measure the status of individual cells, but also to know their spatial location on the original tissue. So this allows us to even understand better the structure of tissues and how cells can interact. So as an application where we look at this, we work with some colleagues studying myocardial infarction, which I guess we all know it's a major source of deaths in our society, and in particular, a process that happens when infarction takes place, which is called cardiac remodeling. And this means that after the heart suffers myocardial infarction, there is a remodeling, which is a change in the structure and the functionality of the heart, which involves processes of inflammation and fibrosis, but it's not really well understood at the molecular level. And then to shed some light on this, we work with colleagues in Aachen, Rachel Kaman Costa, and Milton Weinhausen here in Germany to characterize samples from patients that have suffered infarction. And so we have multiple samples for each patient, and in some cases we have measurements in the ischemic zone, so where the infarct took place, the remote zone, which is like healthy tissue far away from the infarct, and intermediate border zones, and we also have fibrotic tissue. And then we combine single cell technologies like single cell RNA-seq, also technology called ATAC-seq, which tell us about chromatin accessibility, so how open is DNA and transcription factors combined to it, And one of these spatial technologies called Visium, where we can measure many spots in the tissue, and this allows us then to get this spatial results information. Although this technology in particular has the limitation that you don't have the single cell resolution. So each of these spots where you measure RNAs contains a few cells, typically 5 to 10 cells. So then you need to try to understand each spot which cells are present. So then our idea was how we can combine these single cell technologies and spatial technologies by using computational methods, computational modeling, to better understand the interactions between or the regulation of both the intracellular and intercellular processes in the context of myocardial infarction compared to healthy tissue, but also to the chronic disease heart. So the first thing that we did is to use technology called cell-to-location to combine the single cell RNA and the spatial data to estimate in each of the spots of the spatial result data which cell types are present. Because I told you before, this technology doesn't give you what's happening in each individual spot with individual cell, but using the single cell data, we can estimate the presence of different cell types in the spot. And when you do this, you get these results. So here is in a UMAP, in a two-dimensional plot, summarize the profiles of the different samples, and they're color-coded according to whether they are ischemic, so coming from infarcted heart, from a fibrotic heart here in blue, or myogenic, more or less healthy samples. And what you see, as you expect, is in the healthy tissue, you have mostly cardiomyocytes and reds, the major cell type in a healthy cell. As the cell becomes fibrotic, you get fibroblasts, so the major drivers of fibrosis, and in the ischemic heart, the heart that suffer infection, you also see a lot of myeloid cells, different immune cells. And it's also known that in the case of infection, there is a very important immune response. But we want to go beyond looking at cell types, and here's where we again leverage this idea of prior knowledge and the tools I showed you before to try to extract mechanistic insights. So what we did is to apply on each of the spots in the tissue these methods to estimate activity of pathways. For example, here is a patient who has chronic infection, and then we see that TGF-beta, which is a pathway involved in fibrosis, is very active in certain zones of the tissue. Then we can look at the downstream transcription factor activities, and also at the expression level of regulated activities. And if you're interested in regulation, here we could use this attack data, so this information on the how open is the DNA, the chromatin, to better estimate the activity of transcription factor. But then what we wanted to do next is to go beyond looking at what's happening in the spot, but trying to better understand the interaction between the spots. So can we, given that we have spatial result data, can we use this to better understand how particular cell types influence different cell types? And for this, we developed a multi-level machine learning model called listing, which allows you to do that. So it allows you to extract from a particular spatial results data set how much of the changes in a particular spot depends on this spot, how much depends on the direct neighbors, so let's call it the local view, how much depends on more distant neighbors, and so forth. And this framework is flexible, so you can apply it to other type of questions or other type of so-called views. So the effect of different type of structural features. And so when we apply this to this hard data, what we found is that the presence of a particular subtype of fibroblasts, so denoted here with fibroblast 2, is highly dependent of the presence of a macrophage, which is SPP1+. It's a particular macrophage that is known to be involved in autophagy. So, and this was something that was estimated from the analysis of this machine learning method of listing. And here is the underlying data supporting this on spatial transcriptomics. And then this was validated with another technology that does give us the singles resolution, and where we could really see that there is this co-localization of these myofibroblast and macrocytes. And again, this opened the way to more mechanistic studies that we are looking at now, trying to better understand how these macrophages and these fibroblasts communicate. And as I mentioned, it's not involved, or it's thought to involve autophagy and other molecular processes that we are not looking at. Okay, so actually at this point, I'd like to maybe check quickly if there are any questions so far. Also, because since I cannot see you so well, I wanted to see if you could follow, because now in the second part we will change a bit gears in the type of approaches. Is there any question on this part or any comments? Online, offline. Maria, please. So Maria commented in the chat that she has a question. You?
Oh, hello.
Hi, Maria.
Can you listen to me? Okay, yeah. I didn't know that I can speak. So thank you for the explanation. It was very inspiring for me. I wish I could listen it a couple of years ago. But I have a question regarding OmniPath. Do you know if you can select the cell type that you want to build the network? And because you know that sometimes having experimental information or data is not common, at least for us. So then it's difficult to adapt this OmniPath, I think, for a cell type if there is not something that you can select this cell line, or even for a disease, I think. Yeah. If there is this option in OmniPath?
So this is a very good question. So as Maria points out, normally we would like to have information which is cell type specific, but most of the resources of pathways or networks, they are not cell type specific. And so one way to try to make them specific is to use omics data, for example, proteomic or transcriptomic, to refine or to prune a general network to make it cell type specific. For example, you can take data from the Human Protein Atlas or GTEx and then make a network specific. And this is something you can do from OmniPath. I mean, it's not like the core of OmniPath, but it's in different scripts or functions. You can do this, and maybe if you want later, if you send me an email, we can give you some pointers. But it's a very good point. No? In general, the knowledge is not specific, and then we need to find ways to make it more specific. And with all the large omics public data sets, I think it's something that we can at least try to approximate.
Oh, thanks.
Thank you, Maria. So if there are no more questions, I will continue on. As you will see, it's different type of approaches now. Let's see how it goes. So this was my first slide, where I was telling you that we try to use biological knowledge to help the more statistical machine learning model to extract features from omics data. And I hinted in some cases, and we have in different contexts also evidence that this mechanistic knowledge really helps machine learning strategies, but still is far from perfect. There is large room for improvement. We have also looked at this not only in our own research, but we've been involved in crowdsourcing efforts, so-called Dream Challenge, where what you try is to ask the community to develop new methods. So instead of trying to find the best strategy, for example, to predict drug response, you would put data set out there and ask people to make their own predictions, and then you see how well they do. And when you do this, then you learn about many strategies. So this was very helpful. But then also we found that in general, teams that use biological knowledge, like networks, pathways, and so forth, did better than those that did not. But also there is a lot of room for improvement. And that is why we think it's important to complement this very large omics profiling with machine learning, with more focus, dedicated dynamic mechanistic modeling. And these two things, we shouldn't see them as adversarial or decoupled, because the more focused work in mechanistic modeling can be informed by the analysis on the more unbiased large machine learning data. And it also relies, as you will see, in prior knowledge and data, but the key difference is to build these dynamic models. Dynamics means they describe how things respond to perturbations. So you need data where you perturb cells and follow things like that. And so to build these dynamic models in our group, we focus on using a logic formalism. So it's a very simple description of networks, of models, by analogy to electric circuit, but because they are very simple, they're very scalable. But you can use different mathematical formalisms. So often the logic models are described as Boolean systems, so binary systems. But you can use other formalisms, including differential equations to have fully continuous dynamic models. And the other idea then is that you combine, again, the biological prior knowledge, for example, from Omnipath, with experimental data, in this case, different conditions reflecting different perturbations. And we have a tool, CellNode, or variation of phonemes, that builds this logic models out of the data and the biological knowledge. And this strategy to build models, you can apply this to many different types of data. We focus on signaling. So for signaling, the most informative is proteomic and phosphoproteomic, because the signaling is mediated by proteins. So we started with using it on antibody-based bulk data. Then we also expand this to mass spec data. We recently applied it to single cell data. We'll see that in a moment. And you can also use it to study interfaces between signaling and metabolism, combining proteomics and metabolomics. So this is one example of how this applies. If you have mass spec data, if you have mass spec phosphoproteomic data, you can build these dynamic models, and as you can see, they can get quite complex if you measure a lot of phosphorylation sites. In this case, around 800, 800 phosphosites. The main idea then is, can we use these dynamic models to help in this question of prediction of drug response? So can we go back to the large data sets on drug response and try to use these dynamic models to go a bit deeper into why some drugs work or don't work? So then we did that in a couple of contexts. So the idea is, as you remember, we have these large data sets with many cell lines to do with many drugs. So we focus on a particular group of cell lines for which we cannot really predict very well the efficacy of drugs using machine learning, and we do this deep dive with perturbation data and this dynamic modeling. So by perturbation data is you take the cells, you stimulate them, you put drugs, you put ligands, and you measure the phosphorylation of proteins over time. And then you use this logic modeling framework to take a generic network and this to cell type specific data to be cell type specific models, which, by the way, is one way to do what Maria asked before of how we can take a generic network and make it cell type specific. In this case, we do this by doing these dynamic models. And then in one application, we look at colon cancer data. In this case, we use phosphorylation technology called Luminex. We had 14 different colon cancer cell lines from the large screening panel. So we can measure phosphorylation after the perturbation and taking a large signaling network, which is generic, and by fitting it to each of the cell lines, we could get cell type specific models. And then we try to correlate the features of these models to the drug response data and try to see if parameters of the model, and again, this model is based on perturbation data, it's a dynamic model, if features of these models are correlated with the efficacy of drugs. And so this is just to show you the different models. These are the 14 cell lines, and each of them has a different model. The black lines are the lines that are active, strongly active in a particular cell lines, and the light gray are those that are not. So, if you look a bit, you will see that some pathways in this network are active in some cell lines and some in the others. And then if you try to correlate, as I said before, the parameters of this model with drug response, we found multiple cases where there was a strong correlation between the parameters and the drug efficacy. So here is one example where we found that a parameter that regulates or describes the activity or responsiveness of a kinase called DSP3, it is very correlated with the activity of an inhibitor, a MEK inhibitor. So each of these dots is a cell line, and what we see is across the 14 cell lines, the more active is this DSP3, the higher is the SC50 of the MEK inhibitor, which means higher SC50 means less efficacy. So then the hypothesis we had is, okay, if this correlation is also reflecting a causal mechanism, so basically a relationship between the status of DSP3 and the activity of these MEK inhibitors, which are in different pathways. So now MEK is in the MAP kinase pathway, DSP3 is in the ATP-mTOR pathway. So if there is some crosstalk between them, and this crosstalk is important for or affects the activity of the drug, what we reason is it does causal by blocking the drug on DSP3, by making it less responsive to stimulation. Then this would affect the efficacy of the inhibitor, and therefore the cell line should kind of come to this part of the diagram. So we did this for the 14 cell lines. I will only show you the extremes. So as a control, the cell lines here is already very sensitive to MEK. If you put inhibitors of DSP3, we have different inhibitors. As you put more inhibitor, the efficacy of MEK inhibitor does not change, as you expect, because it's already very efficacious and it's already top. But if you go to the cell lines in the top right that are not sensitive to MEK, by adding the DSP3, you increase the activity of the previous drug together, meaning that indeed these drugs together work better than the MEK inhibitor alone. And that's one example of how you can use a dynamic model to find first a model biomarker that you will not see only from looking at omics data. So in this case, this activity at DSP3, we found that it's predictive of the efficacy of the MEK inhibitor, but also it allows us to suggest a combination of two inhibitors, one MEK, one DSP3, that are together more effective than the drug alone. We then did a variation of this study using another technology called mass cytometry. So in mass cytometry, you measure the phosphorylation of around 30-40 proteins at single levels, in single cells. So this means that you can get a lot of cells on individual cells, but then we apply the same strategy. This was done with the lab of Baldo Miller. And in this case, it was not colon cancer, but breast cancer, but it's the same idea. So you build from each cell line a data set of perturbations to drugs and ligands. You take a genetic network, you build one model for each cell line, and then you try to relate the model features to the drug response. And here is to show you the model. So as I said, this data is with single cell resolution. So you really have a lot of data points. So in total, you measure over 80 million cells. And then you have time courses of how all these different cell lines respond to stimulation with drugs and so forth. You take some up to build these logic models. In this case, using the differential equation, so you can really capture nice dynamics. And then this is just to show that the models we built could look at the differences in different cell lines in the drug response of dynamics. And then this led to biomarkers based on the model, like in the case before, that you could not find by simply looking at the basal data. And so the last thing I wanted to say in this context is, can we apply this idea of dynamic modeling and perturbation data to patients? So the last examples I showed you on colon cancer and breast cancer, they were both in the context of cell lines. And this is helpful and interesting, but at the end, of course, we want to know how to treat the tumor in patients. But to make these perturbation experiments, you really need a lot of material. So you need to take cells, treat them with many drugs, measure the activity. So this is not something that you can do with tissues. The question is, how can you apply these strategies really to patients? If you are looking at a disease in the blood, that's still doable because you have you can get a lot of blood from patients in many cases. So this is a study led by Marty Bernardo. It was published last year, where we look at multiple sclerosis. So we had blood from around 150 donors. And then again, the same strategy. So from these samples, from the immune cells of the blood, you perturb them with different ligands and drugs. You use this to take a generic network and make one model for each of the donors. And then these patients models, by studying them, you can look for combination therapies by trying to find which drugs together repair the circuit of the disease cell. And so in this case, so this is the summary. So we have this large network covering the major signaling pathways of these immune cells, and doing some analysis of the models, we come up with a combination of two therapies. And then this therapy was then validated in the mouse model, where, as you can see, so the combination of these two drugs, of fingolimod and TAC inhibitor, this means the phenotype of multiple sclerosis in a much stronger way than the individual drugs. So you can do this if you have blood, you can generate data to build dynamic models. But what about if you have tissues, meaning organs that are not liquid, which is much harder to extract material in large amounts? So for this, what we did is to work with Christoph Merten, at the time in Vienna, in Lausanne, and unit partners in Aachen, Thorsten Gramann and Neumann, to use microfluidics to perform drug screenings in very small amounts of material. So the idea is that you encapsulate in small droplets, as depicted here, a few cells with some drugs and some information to barcodes to know which drug is with which cell. And then you measure the efficacy of the drugs on the cells using a Cascade reporter, so how much you trigger apoptosis, although now we can also connect this to RNA-seq. But the idea is that by using these microfluidics, as shown here, you can really automatize the process of screening drugs in very small amount of material. So you can take biopsies and do it right there. And this is just to show how you can do this in 4 biopsies from primary tumors and make a lot of pairwise combinations of drugs. So this is 8 by 8 drugs in combination, and you can see the different effect of the drugs. But then what you can do is to also use this data to build models. So again, the same story as before. So you have from each patient a drug response data set, you have a generic network, and then by training the generic network with the data of each patient, you build a model for each patient. And then by studying these models, you can try to come up with better combination of therapies. So this is the model that came out of this analysis. It's a model that explains the regulation of cascade, so the apoptosis, which is the result of our experiments. And then, as before, you find the activity of different pathways, changes across different patients. In this case, it's represented by this squares here. So each of the little squares is the level of activity in each of the patients, and it's pairwise across the patients. But then you can take the models and use them to simulate the effect of drugs. And that's another feature of these dynamic models, that it's not only that you can look at them, but you can simulate new conditions. Now, what happens if now I put a drug here, I put a drug there? And so we did that, and in particular, we tried different drug combinations. We tried to see which drugs can be efficacious, and we could not go back to the patients, but instead of this, we did the same exercise in cell lines and now xenografts, and in those cases, the model predictions, when taken back to the cell lines or to the mice, the combinations predicted by the model were efficacious. So this means that using this perturbation data from this microfluidic technology combined with mathematical modeling, it's a good strategy to come up with new drug combinations. And as I said, we couldn't do this in patients, but this is not something we are starting to do. So we're going to start a trial, trying different combinations on different patients. And this is just one example. So it's, of course, only one patient. So this doesn't prove anything. We need many more patients to really see if this strategy is really helpful. But so in this case, as an example, so this is a patient whose treatment failed. So the standard of care didn't work, which is a combination of these two drugs. So our colleagues in Aachen took a biopsy. This microfluidic experiment was done. And from the microfluidic, this different combination was proposed of cisplatin and gemcitabine. So if now you go to the patient who had this tumor that has, as I said, not responded to the first therapy, then you give this combination, and then the tumor largely decreased, which means that the drug is being effective. Of course, this, as I said, this doesn't prove anything, or even in the case of this patient, eventually the tumor developed a resistant mechanism and came back. But this is an illustration of how we think that we can really, in the near future, using microfluidics and mathematical modeling, perform drug screenings directly ex vivo from patients. And this information complements the more standard information that the pathologists and the doctors use to pick treatments, because it's a bit more functional. It's how the cells respond to the drugs. And hopefully this can help to improve how we treat cancer patients in different conditions. So with that, I'd like to finish. First, I'd like to thank the people who did the work in the lab, in particular. So the NES is the main developer of OmniPath. Rosa did all the Carnival work on the cell lines. Aurélien did the multi-omic integration Cosmos. Martin did the multiple sclerosis study, and Federica, the last ones on cancer with microfluidics. By the way, we are looking for postdocs and PhD students in case any of you are interested. And Heidelberg is a nice town, maybe not as nice as Barcelona, but this is not nice, small research town. And I'll just try in the last few minutes to summarize what I tried to tell you today. So I think that there's a lot of value in combining, let's say, machine learning and data analysis with more dynamic modeling for modeling of patients, of organs, and for disease pharmacology. First, machine learning, if you apply this to omics data, as I tried to state, is certainly a powerful strategy, but still is a long way to go. And we think that combining or using biological knowledge helps a lot these type of strategies, because you increase not only the performance of machine learning, but also the interpretability. In this context, I emphasize the value of this idea of the footprint as a way to extract features from omics data. So if I want to know about a process, I look at the downstream genes, for example. Then I talk a bit about how these strategies can also be applied to single cell and spatial data, which is very exciting because it opens new opportunities, but also has some challenges for the computation. And the last part, I describe how we can use dynamic models of signaling pathways based on perturbation data, and how this can many times shed light on things that the pure machine learning couldn't tell us much. And this is mostly because we think of this idea that how cells respond to perturbation is very important of its functionality. And this cannot be captured only by profiling in steady state, in the basal state samples, but seeing how they respond to perturbations. And so based on this, in general, our idea is to have biological knowledge, a genetic model. This model, you can then train it to patient-specific data, can be this static data, the machine learning strategy as we talked at the beginning, or can be more dynamic data, data after perturbation. But in both cases, we try to build a patient-specific model and use it to extract mechanistic insight into the disease, but also to better predict efficacy levels. And with that, I'd like to finish also saying again that all our tools are freely available, and I'm happy to take any questions. Thank you for listening.
Thank you very much, Julio. So if there are questions, I will bring the micro. Questions also in the online?
Maybe I can break the ice. Well, thank you very much, Julio, for this very, very nice talk, and it's really impressive and inspiring work. My question is really a curiosity. So in a project that is supporting this summer school, we're targeting musculoskeletal tissue regulation with very long inertia of tissue changes over several years. And during this process, there are many modifications because of mechanical loads and cell senescence. There are many modifications of the cell sensitivity to its microenvironment. So the interplay, for example, between inflammatory factors and the chronic mechanical loads that the organ is seeing, so as becomes modified over the time. And this is what drives, in the end, the phenotype. So I understand this is an extremely complex question, but how do you envision this could be tackled through modeling, for example, by using the kind of approaches that you've presented?
Yeah, it's a good question. So I guess if I understand it, the major challenge would be the mechanical aspects, no? I mean, let's say what I described focus on molecular interactions, communications, but as I understood your context, there is mechanical forces to include into the model. Is this right?
So to put it a little bit more biological, let's say that you have mechanoreceptors on the cells that have a specific response under a specific biochemical environment, so with specific cytokines. So at time 0, the response is one thing, and at time 10 years, the response becomes another thing. And so there is a whole history that is actually important to drive a specific phenotype. And this is, for example, in osteoarthritis, this kind of disease, that's very important to capture that in order to have proper drug treatment strategies, which we don't have right now.
I understand. Okay. Yeah, I guess it's a challenging question. I mean, one way you could start thinking of this could be, as you said, that over time, based on the environment, how the mechanoreceptors respond changes. So maybe the simplest approximation could be that you have like different snapshots, right? So you could envision that over time, as this effect of the environment comes in, the kind of the wiring of the mechanoreceptor network changes. And I mean, this is a bit, could be analogous to, for example, how the network of a patient in cancer evolves over time. So that could be one way, very simplistic snapshots. And then next step could be to basically have kind of a dynamic model that describes evolution over time of the network. I mean, we haven't done this ourselves, but that would be how I would think about this. But yeah, it's a very good question.
Hi, thank you. It was very nice seeing Luminex being in the presentation. So I want to ask, aren't a little bit the networks biased towards how many antibody pairs you have when you build the networks? Wouldn't it be better to have less cell lines and do mass spec data and get phosphoproteomic data from mass spec so you can have a bigger network and you have better networks?
So that's true. So and in a way, it's a trade-off. No, so the mass spec, although it's improving a lot, still it takes more time and cost than running the antibody kits. So we have done that as well. So we have projects where we have mass spec data with phosphoproteomic, and then you have much larger networks and more unbiased. But then you cannot do, at least in the same budget, you cannot do so many conditions. So it's a trade-off between coverage and not missing out things that you know about, and exploring the dynamic response. And one could think, I showed you one extreme, we have done the other extreme, you could also think of doing hybrid. You could say, okay, maybe I do first mass spec with few conditions just to identify, but well chosen, for example, drugs that I'm most interested in. This is, and build network models, and you can build these models, right? So I think I showed it briefly. So you can build such models for mass spec, like this one here, right? So, and then use this to see, okay, which of the processes seems to be key, seems to be responding to my drug of interest, and then maybe if I want to do more conditions or more time resolution, then maybe go to the antibody. Like in this case, I would see, I don't know, ERIC is very important, and ATT is very important, pKa, and then I follow up in a more detailed manner with nodes. But there is always this trade-off, coverage and throughput, which means throughput means, in our case, more drugs, more ligands, or more time points, which is another key aspect. The time point at which you measure for dynamics is essential, and I didn't discuss this, but if you have only one or two time points, you have to choose them really well, but they inform you about how the cell responds to stimuli. And that's again a trade-off, coverage and picking the right dynamics.
And then another question, because when you build networks, and then you see that hub proteins get the more. When you have a condition, usually it's a hub protein that connects. It's a connector in the network. So can, for example, the networks first highlight proteins that they're not hub proteins, and maybe also so what kind of drugs they're not gonna be bad for the organism? Because okay, you can find a protein, but if it's pretty much like a napalm in the organism that kills everything, then it's not like a drug.
Yeah, you're very right, and in a way, what you look for is differential activity, right? So you should always compare against the effect on a healthy cell and try to see something that affects what is very different. So, and it's also what you said. So if your target is a very active protein, it's very likely to have a lot of side effects, and you should take that into account. Also, the hubs, this is also a bias that comes into the network. So when we draw these networks, like here, you have ATP linked to many things. In truth, we don't know if this is really because it's always very important molecule or because we have a lot of knowledge about it, because it's been studied. And we look at this recently, and there are clear biases in what the literature focuses on, right? And then again, this will propagate the models. And in the models, you are more likely then to find the hubs because they are hubs in the network, which means they were very studied. So it's a bit of a self-fulfilling prophecy. And there are ways in the algorithms to try to correct it, to kind of say, okay, I kind of normalize against the hubs, and I only find the hubs if they are really more important. But yeah, these are very good questions, and we always have to keep in mind, on one hand, these biases in our knowledge, and second, as I said, the fact that trivial solutions to stop the growth of a tumor is, of course, a poison that may also kill the patient. Yeah.
Yeah, and maybe a bit related to that, so especially when you're working with knowledge-based modeling, so you're not obligatorily controlling very well what you should include, what you shouldn't include, and then there is a huge work of network cleaning that is based on testing, different tests, whether you get an expected answer according to a specific perturbation. And within the kind of huge networks and systems of networks that you're handling, so do you have a standardized way actually to do this cleaning? Are you successful in defining standardized tests? And is it necessary actually to go for the development of these standardized tests if it doesn't exist?
So what would you mean by a test?
A test means that you perturb the network in a specific way and you're expecting a specific response. So a benchmark, which allows you then to say, okay, this node is a bit annoying, and I will get a little bit more into that, and effectively it shouldn't be here or it shouldn't be connected that way.
Yeah. Yeah, this is a very important limitation and or question to keep in mind. And I think a limitation is often to have really ground truth. So how do you know what should come out? This is, I think, general actually many of these strategies, and I mentioned this when I was talking about cell-cell communication. But for the networks, I mean, you can indirectly test that the models are working well. If, for example, you leave out some data and you try to see if your model can predict the data. This can be particular condition, like a particular knockdown or condition that you have done, or can be leaving out some of the measurements. In this case, see if the model can predict some of the things that are going up in red. And that's very important to get a sense that the model is working. Yeah, as you were saying, so if you can use the model to make a prediction on the importance of a particular node, and then follow this up experimentally and block it, and see if it really is as important as the model says. But yeah, so in a way, you can use the models to make predictions and see how well the predictions work. But truly knowing what is the underlying molecular ground truth, I think it's harder, at least at large scale. And that's why, yeah, we have to go with indirect.
Well, actually, I have done another question. So, how is the level of translation of all what you're doing to clinics? So, can you envision that at some points this can become a part of routine exploration, advanced diagnosis, advanced assessment of fitness of a patient to specific drug or combination of drugs? How are you seeing that, and then what might be the challenges?
So the first part, which is ways to extract features from omics data, this we have worked a bit here with people in the clinic in Heidelberg that they have trials where they're doing genome and transcriptome profiling, and they run some of our tools, and you will see if they really add value. But I think if that's the case, no, that's a relatively easy way to contribute, because if the data is there and it will generate it, running some of these methods on top is not that hard. And then, of course, this data or this information has to be combined with others in the tumor boards, let's say. So the latter part, the dynamic modeling and the perturbation data, that's much harder, because as I saw at the end of this microfluidics, you really would need to generate dedicated data. And we would like to move in that direction, and we will try, but you need to develop basically a device, a system that can be run robustly in a hospital, right? So So one thing is to do the experiments in a research laboratory by really experts, and the other thing is to be able to run such perturbations experiments routinely. And I think this needs technological development. We are optimistic that at least it's worth the try, but this will take a bit longer time.
Thanks. And maybe the last question, Baldo will kick me out from the auditorium. So here there are several people working with population cohorts, specifically twins UK cohorts, so that has homozygote twins, and the Northern Finland birth cohort. So how do you because I hadn't the impression that you're working actually with population cohorts in order to make the development, or maybe I'm wrong. Do you think these cohorts bring an added value to all these developments and maybe also the testing and applications?
So you mean cohorts of patients, like or population cohorts, like?
No, not patients, actually. People from the general population that are recruited and then followed up over several years. So we're talking about decades, and they have regularly blood samples, urine samples. They also might have different medical imaging explorations. And so you have actually the whole history. So some patients will stay healthy, some others will develop certain conditions, and so these are the population cohorts I'm talking about.
Sorry, yeah, like UK Biobank, for example, no?
Exactly, yeah.
Yeah, I mean, we have looked a bit in that. I mean, I would say we are not really the experts, but I think these are really, really, really great resources because, yeah, as you said, it gives you a more natural population that reflects the development of the population, and as you said, is traced over decades, and that's very important, I think, for many diseases and can be used for many questions. And I think it's really helpful that also the UK Biobank make this data available to any researcher, right? And you can come, studies like ours, but many others, and try to see what you can learn. And as I said, in some specific cases, we are looking at this, and also these resources will keep increasing. More patients, more molecular layers. I think they will become a super valuable resource, and computational groups like ours, can have a lot of fun looking at this data.
Thanks. Sure.
A question regarding the study that you were doing, that you had many patients and that you built an individualized network for every patient. Out of curiosity, were there any, for example, if you saw the network of every patient, could you, for example, say his age, if he's smoking, BMI, comorbidities, just from the network? Could you see if there are underlying hidden patterns just from what is phosphoproteomically there?
So I think that makes a lot of sense to me. Probably we do not have enough amount of patients to get the signal out, because I guess the signal would not be very trivial. I mean, going back to the UK Biobank, I think in that place is where you have a lot of patients, you can really see this model. But if I had to bet, I would say there is a signal that you could see in the models and in the phospho data, but you will need larger cohorts to really see this in a significant manner.
No? Just take mine.
More questions? Okay, so maybe we can move to the next. Thank you very much, Julio. Nice seeing you.
Thanks, everyone. Thanks, Baldo. Bye.